again, it's my great pleasure to have Professor Simone DI GIOVANNI to visit us and also give a lecture. So Simone did his MD and also PhD training in Italy, then moved to US, Georgetown for his PhD postdoc training. Then he started his independent career and at Germany, Tübingen, then moved to UK. So he's currently a head and also the distinguished professor at Department of Neurology and Division of Neuroscience in Imperial College of London. And he has done fantastic work in both the axon regeneration and also the metabolic changes and interaction with immune system in different conditions, including traumatic injury and also diabetic, etc., in the nervous system. So I hope he actually he's also a clinician. So he's a very unique background, both in the basic research and also the clinical practice. Hopefully, will give us a very unique perspective in this control mechanism of the axonal regeneration from metabolism to neuroimmune interactions. Okay, so I hope you guys enjoyed the lecture, and without further ado, I'll give the floor to Simone. Okay, let's have.
Thank you very much, Kai, for the kind invitation, and it's great. I've been in China for 2 weeks, and it's been very, very interesting, and the first time, and it's great ending it here before going back to London tomorrow. All right, so I will start by showing you, I guess you are from a diverse background, so a very brief introduction that I think will make the point clear. This is an injured spinal cord of a mouse. In this case, you can see the sensory neurons that convey sensory information to the brain are damaged. The axons are damaged. They do not regrow. Similarly, the motor neurons that come from the brain and innervate the muscle for muscle contraction are also damaged following spinal cord injury. They collapse and they do not regrow. Trying to enhance the growth capacity of these axons is essential to allow reconnectivity that will limit disability, neurological disability, and hopefully enhance sensory motor function. The other condition that we're interested in is actually equally dramatic and even more frequent than spinal cord injury, which is peripheral nerve damage. This shown here is a sciatic nerve crush in a mouse. You see that following sciatic nerve crush, you have some level of spontaneous axonal regeneration. However, it's not dense enough and it's not quick enough to reach its target that can be the muscle or the skin or other organs. And therefore, injuries in the peripheral nervous system also lead to permanent neurological disability. There are many ways that one can approach the problem, and what I will show you today is mainly the crosstalk between immune cells and neurons and how that can affect their regenerative capacity. Additionally, I will show you how some metabolic states can directly affect neuronal biology or indirectly affect neuronal regeneration via changes in the immune system. In the last several years, my lab has been interested in understanding environmental variables, broadly speaking. They include activity-independent changes, dietary changes, and age. Because, of course, these are essential variables that our life has to deal with. In the interest of time, I will not talk about some published work, but I will just talk about one recent published work and 2 unpublished stories that you will see they are interconnected conceptually, although they are independent from one another. However, we'll briefly summarize 3 modes of neuroimmune interaction that my lab has defined in the last few years, which I have all the common denominator to be focused on the regeneration of sensory axons. The first one that I want to briefly summarize is a paper that we published last year when we showed that CD8 T cells that are typically cytotoxic cells, but they can also be immunoregulatory, are attracted to the dorsal root ganglia, which are the structures that convey sensory information from the periphery to the brain, during aging. And they are attracted to the sensory root ganglia because in neurons with aging, there is the overexpression of a chemokine called CXCL13, which attracts CXCR5, the ligand, CT8 positive T cells to the dorsal root ganglia. After injury, the neurons express MHC class 1, which is recognized by these cells, and as a consequence, there is regenerative failure. We were able to take advantage of a humanized monoclonal antibodies that antagonize CXCL13 to promote regeneration and recovery in these animals. So this is a mode of interaction that's a receptor ligand CD8 T cell dependent interaction that is directly links CD8 T cells with neurons that is reversible with monoclonal antibodies. A second mode of interaction that we published a few years back has to do with the extracellular vesicle mode of communication between macrophages that proliferate and are activated following a sciatic nerve injury. These macrophages release extracellular vesicles that contain NADPH oxidase, which oxidizes specific proteins in the axons, including P10, which is a very well-known inhibitor of axonal regeneration. P10 oxidation activates the regeneration program of sensory neurons. Later, we also shown that this overexpression of this enzyme after a spinal cord injury is able to promote regeneration and some level of recovery in the central nervous system. A last mode of interaction, which I will now actually discuss with you in more depth, has to do with the secreted factors. Secreted factors that in this case are released by neutrophils that are activated, you will see now, by a gut metabolite called indole propionate to support axonal regeneration. I will now tell you this story, but before going there, I have to tell you how things started, how we got there. And that story, in a way, started a few years back when we investigated whether environmental enrichment, that means putting animals in an enriched environment, as you can see here, versus an impoverishment, which is standard environment, which is kind of impoverished environment, if you will, because it was known already that environmental enrichment enhances learning and memory and synaptic plasticity in the brain. Since some of these mechanisms at the molecular level are recapitulated in axonal regenerative conditions, we investigated whether environmental enrichment could enhance signaling in the sensory neurons that would support axonal regeneration. Long story short, we published this paper a few years ago. We found basically that environmental enrichment activated neuronal activity, activated a molecule called CBP, which acetylates histones that promotes the regeneration program. We could then use a small molecule that activates this protein called CBP following spinal cord injury, and that promoted the regeneration of sensory fibers, and not shown here, but published already, also motor fibers and functional recovery. All right, so this is the background to say that environmental enrichment-dependent mechanisms that contain as a major component exercise are shared when with dietary changes. So that led to the hypothesis that if we put animals in a dietary regimen, such as, for example, intermittent fasting, maybe we could identify metabolic-based mechanisms that we could leverage to enhance the axonal regenerative ability. So to this end, we did a series of very simple experiments where we put animals in a diet, normal diet, AL stands for ad libitum, or IF, intermittent fasting. This means for 10 days or 5 weeks initially, the results were identical between 10 days and 5 weeks. Therefore, we focus on 10 days because it's just faster. And then, so we did feed, no food, feed, no food, okay, for 10 days. Then we did a sciatic nerve crush, and we waited 3 days, 1 day, or 3 days, or longer. But anyways, now you will see 3 days as a model system, and we measure regeneration. And we found that intermittent fasting enhanced axonal regeneration. The first thing we did then to try to understand why was metabolomics from the serum. I thought it was a logical step because clearly intermittent fasting changes the metabolism. And what we found was a number of metabolites that were differentially expressed between ad libitum and intermittent fasting, and included some top metabolites that were either exclusively produced by gram-positive bacteria or produced both by the endogenous system and gram-positive bacteria. For example, IPA, indole propionate, I will talk about it later, is exclusively produced by gram-positive bacteria. Anyways, overall, the top metabolites suggested that intermittent fasting was altering the gut microbiome. To have a first evidence that this was the case, then we took the feces from the animals that underwent intermittent fasting versus ad libitum. We fed animals that had ad libitum diet via gavage, or fed them with animals that from the feces from intermittent fasting, and we compare axonal regeneration. We found that if you give feces from intermittent fasting animals to ad libitum animals, there was an enhanced axonal regenerative capacity. Although this was modest, probably containing things that were potentially useful, others that were less useful. However, this gave us a proof of principle that there was something in the gut microbiota that could enhance axonal regeneration when the animals were on intermittent fasting regimen. Since I told you gram-positive metabolites were on top of the list, we depleted the gut microbiome, gram-positive microbiome, with vancomycin for 5 days according to classical protocols. Then we put animals in intermittent fasting or ad libitum versus control. And again, we measure axonal regeneration following sciatic nerve crush. You see in red, intermittent fasting enhances axonal regeneration. Vancomycin in the other groups blocks the axonal regenerative ability triggered by intermittent fasting. Then we did metabolomics from the serum again. And this was a very useful experiment because the only metabolite that was blocked by vancomycin in both conditions, and still, of course, induced by intermittent fasting, as confirmed earlier, was indole propionic acid. Here on the right, you just see more targeted metabolite measurement of indole propionate, that is more sensitive, of course, than just general metabolomics. But these are essentially the same data. Okay, what is indole propionate? Indole propionate is a metabolite of tryptophan produced almost exclusively, you see later a piece of data that actually suggests it is true, by Clostridium sporogens, which is a gram-positive bacteria. Then I found in the literature that a group at Stanford had produced, for other reasons, Clostridium sporogens that do FLDC. They have a metabolic impaired pathway in the production of indole propionate. So we received these bacteria, either wild type of mutant bacteria that do not produce IPA, to test, and we depleted the microbiome, and we gavaged these bacteria, wild type or mutant, to test whether indeed the bacterial produced IPA supported axonal regeneration. And you can see here, that's what I was mentioning earlier, the IPA in serum, after you deplete the gram-positive microbiome, is basically undetectable. And when you give the wild type or the mutant, again, you see clearly the IPA in the serum comes only from this bacteria. And this was done in collaboration with the Weizmann Institute. That, the metabolite measurement was carried out by them. And axonal regeneration was reduced in case of lack of production of IPA. So this suggested directly that IPA enhanced axonal regeneration comes from bacteria. Gut bacteria are in the gut. However, when a metabolite is in the gut, typically eventually goes also into the bloodstream. So the question now was, is IPA produced in the gut essential for regeneration, or is its systemic presence important for regeneration? In other words, is this an indirect effect, or is it direct effect from the serum that might then affect the dorsal root ganglia neurons? To this end, we did two experiments. We either gave IPA by oral gavage, so working through the gut, or systemically. And we found that in both cases, IPA led to the similar extent of axonal regeneration. I'll just show you one of the two examples. This is gavage of IPA that enhances axonal regeneration. This is the concentration of IPA following oral gavage. Therefore, this suggested that bloodstream presence of IPA will mediate axonal regeneration. To understand downstream mechanisms, we did sequencing from dorsal root ganglia following IPA before and after an injury. Okay, we can jump that. And what we see here is that IPA delivery enhances neutrophil chemotaxis. And following injury, again, we have cellular response to interferon, inflammatory response, and so on. So clearly, the top categories that are IPA dependent were related to immunity, and the top were pro-inflammatory signaling, including coming from neutrophils. Interferon gamma can come from both neutrophils and T cells. And why we didn't see any change IPA dependent in the sciatic nerve or in the dorsal root ganglia in different immune cells, we found the neutrophils were increased in the perivenular space following IPA in control conditions, IgG, and neutrophils were blocked when we used an antagonist of CXCR2 that we found overexpressed in our RNA sequencing screen, and then we found expressed in DRG neurons, attracting neutrophils into the perivenular space of the DRGs. They don't go into making a contact with the DRGs. Neutrophil remains in the perivenular space, and they're known, of course, to release a number of factors. Then, of course, in the same experimental design, when we antagonize neutrophil chemotaxis down here, we block the IPA-dependent increase in axonal regeneration, suggesting the neutrophils are important for IPA-dependent phenotype. Now, in the published paper, if you're interested, you can look at some downstream mechanism. However, I will, in the interest of time, now move on to the last part of the story, where we used IPA following a sciatic nerve crush. And what you see here is there is an increase in epidermal innervation of the sensory fibers, as shown here, which is quite significant. There's acceleration of recovery of sensory fibers that sense thermal sensitivity. And while there is no difference in the bone fray, which detects allodynia, which is a form of mechanical pain. So in summary, IPA, which is not the beer, unfortunately, enhances axonal regeneration in the peripheral nervous system. I think actually Kai gave, did you give IPA into the optic nerve? Or?
We tried. But
yeah, but there was no difference in axon.
Only do injection, right?
Injection.
I don't know how much that can sustain.
Yes.
Maybe some kind of, maybe we can look into.
Yeah, anyways, maybe this does not work in the optic nerve. And however, in the peripheral nervous system, had an increase of axonal regeneration mediated by neutrophils. I mentioned the optic nerve because it's quite interesting because a few years back, there was a paper from Benjamin Segal and Roman Geiger that showed the neutrophils, and they actually studied neutrophils in more details than we did by single-cell sequencing, I believe. At least a subset of neutrophils can support axonal regeneration in the optic nerve. But I think in that case there was an inflammatory activation, and so it's possible that in the optic nerve maybe you need some combination of treatment to enhance IPA-dependent regeneration. What is attractive about IPA is that it's a metabolite that is physiologically present in the serum. Actually, it's the highest gut metabolite published in human serum, which is quite interesting. You can easily buy it, and I'm not suggesting to take it, but it could support clinical translation. It's not going to make the difference alone, but could be part of the cocktail to enhance repair. All right, so you can imagine how intermittent fasting is a situation where multiple mechanisms coexist. And while we identified the bacterial-dependent mechanisms, we wanted to better understand what could be endogenous mechanism that would support the axonal regenerative ability, and perhaps we could learn more and leverage upon that to enhance axonal regeneration. To this end, we did dorsal root ganglia RNA sequencing of the whole DRG. So this is not single cell, to look at the DRG microenvironment. We did it a few years ago. Probably now we would do single cell. Anyways, in intermittent fasting versus ad libitum, same identical experimental paradigm, 10 days of ad libitum versus intermittent fasting. And when you look at the differentially expressed classes, these are induced genes, you see lots of things that make sense: hormone signaling, lipid, including in sensory neurons, changes in metabolism, metabolic processes, and some of neuronal categories which are potentially involved with activity-dependent growth. If you look here, a number of genes of significance, you see again, lipid metabolic processes. Mainly, if you look into that, it's lipolysis. So it's lipid catabolism, increasing cholesterol transport. And there is a high also down here, significance for circadian rhythm. Indeed, it is known that intermittent fasting, as opposed to fasting, which is a very strong caloric dependent condition, intermittent fasting changes the circadian clock in a way that aligns the circadian rhythm, and it is known to potentially underline something that this data suggests. What this data suggests is that several of these processes, including circadian rhythm and lipid catabolic processes, are underpinned by leptin signaling. Therefore, we wondered whether leptin signaling might be enhanced by intermittent fasting, supporting perhaps axonal regeneration in sensory neurons. Leptin is a hormone which is released by adipose tissue, by adipocytes, regulates our feeding behavior by working through the specific nuclei in the brain by decreasing feeding behavior when leptin is up. And it works in counterbalance with another important hormone, which is called adiponectin. More importantly, perhaps, for axonal regeneration is the discovery, for me at the time, the leptin binds to leptin receptor, which we found being expressed in sensory neurons, which it's autophosphorylation in part, and then JAK-dependent phosphorylation leads to activation of STAT3, ERK, MAP kinase signaling, CFOS, and PI3 kinase, AKT, and mTOR. So if you have any familiarity with axonal regeneration, I would challenge you to find a signaling molecule that does all of this together. It was quite stunning, I must say. And so we could not ignore this hypothesis, I would say. Additionally, evidence in other systems, you see here, in other organs, have shown before the intermittent fasting, although by an unclear mechanism, enhances leptin sensitivity in target tissue. Nothing was known about neurons that are outside of the brain, classical feeding behavior dependent neurons. Okay, so then we first looked whether intermittent fasting enhanced signaling. As I've shown you, phospho-STAT3 is a paradigmatic signaling molecule for leptin. And before injury and after an injury, following intermittent fasting, I will show you just after injury here, in the interest of time, we found an increase in phospho-STAT3 following intermittent fasting. And we found the expression of leptin receptors in DRG neurons, and with appropriate mutant controls, we indeed found the leptin receptor is expressed in these neurons. Next experiment we did, we wanted to activate leptin receptor we wanted to block leptin receptor signaling in intermittent fasting condition to see whether indeed intermittent fasting would trigger axonal regeneration via leptin receptors, via leptin signaling. So you see here, we either deleted with the Cre AV virus leptin receptor in leptin receptor floxed animals, or we use control viruses. We gave 10 days of ad libitum or intermittent fasting, performed sciatic nerve crash, waited 3 days, and bottom line, regeneration index, or there's two different measurements here for axonal regeneration in sensory axons. You see that intermittent fasting promotes regeneration. We knew this already. And when you block leptin signaling, intermittent fasting dependent regeneration goes back to baseline. And we didn't know this actually from a previous paper. Then next, we gave leptin systemically, mimicking a bit what adipocyte release normally would do. So now we overexpress the system, and the idea is systemic leptin will bind to its receptor in sensory neurons, and you can see that in control condition it promotes regeneration, mimicking intermittent fasting dependent regenerative profile. And again, here we deleted leptin receptor in the same way as I showed you earlier. And when you delete leptin receptor, leptin-mediated regeneration is blocked. Now, the issue with leptin, when you give leptin systemically, although I think it's a nice experiment, is the leptin receptor is expressed in immune cells. And immune cells play an important role to control regeneration, of course, in particular in the peripheral nervous system. They do make a difference to regeneration. So we wanted to see whether neuronal leptin could promote axonal regeneration, neuronal leptin signaling. Now, leptin is released by cells and binds in autocrine or paracrine manner to its own receptor. So we could take advantage of this knowledge by overexpressing leptin in neurons and activating neurons themselves by autocrine mechanism. And, okay, sorry, this is just to show you the leptin delivery does activate the signaling. And when you have leptin receptor, which is deleted, you have inhibition of the signaling. So what we did now is give leptin by V virus overexpression in the sensory neurons versus a control. And you can, this is just to show you leptin expression, GFP expression, and leptin. And you see that axonal regeneration is enhanced when you give overexpressed leptin in neurons. Now, to prove the mechanism, before that, I'll tell you more. When you overexpress leptin, you have an increase in STAT3 signal within neurons. And when you wait also, this is probably 3 weeks following injury, several weeks. I'm not positive it is 3, but I think about 3 weeks, we can also see an enhancement of epidermal innervation. So the effect is quite robust. Then we went to more difficult conditions, which is in terms of growth, which is spinal cord injury. These are sensory neurons. Spinal cord injury does not allow sensory neurons to invade the lesion site. And when we give overexpressed leptin, we saw quite a substantial regeneration of these sensory neurons across astrocytic bridges in this crash model of spinal cord injury. And honestly, I've rarely seen this nice regeneration of sensory neurons before. Probably, again, underpinning the fact that leptin, as you've seen from the signaling, it's probably quite a powerful signaling molecule. Good. Now I come to what I was thinking to tell you earlier. And yes, good. So what we did now, we wanted to prove the mechanism, the autocrine mechanism. So while when we tried to measure leptin from the serum, it was very diluted, so it was quite difficult. We did try. We were able, I think, to anyway prove the principle by overexpressing leptin in vivo, culturing dorsal root ganglia neurons, either GFP or expression of leptin, and that promotes regenerative growth as expected in either wild-type animals or leptin receptor, DB, null neurons. When that happens, when we give leptin receptor null neurons, GFP and leptin GFP do not change in terms of regenerative growth, suggesting that leptin, which is given into neurons, works through its own receptor. And so it must come out to work through its own receptor and then bind its receptor again. How does leptin exert its regenerative effect? So again, one step back, systematic approach. Again, in the usual experimental design, we perform a neuronal enrichment. It's a technique that we set up in the lab. Hopefully we'll publish the methodology soon, where we have a strong enrichment of neurons versus other DRG cells that, as opposed to FACS sorting, it's less traumatic and allows many more neurons to be recovered. And what we did that, of course, to try to have an enriched population of leptin overexpressed neurons as opposed to the whole DRGs. And this sequencing was a very fortunate sequencing because, as you can see, it's very monothematic, meaning that you have a strong enhancement of transcription and phosphorylation, which is expected by leptin overexpression, if you see that. And of course, those phosphorylation pathways have been shown to impinge upon leptin. Of course, they are expected to enhance transcription. And then we went to do a Fisher test to see correlation between overexpression of leptin versus intermittent fasting ad libitum. See, this means that overexpression of leptin mimics intermittent fasting, mimics sciatic nerve injury, but does not recapitulate a non-regenerative condition like dorsal column axotomy in the spinal cord. Then, since we saw that transcription was strongly activated, the effect lasted for apparently anatomically for a few weeks, We looked at classical epigenetic marks that we previously shown to be associated with regenerative ability, such as K27 that was enhanced after leptin overexpression in the gene neurons. We also looked at the activation of phosphoserine 2 on RNA polymerase 2, specifically in neurons that overexpress leptin. That's important for transcription, and also that was increased. Finally, the last piece of data, before I tell you briefly the last story, was to prove if indeed transcription was important for a leptin-dependent mechanism. So we use a classical approach that others have used before, which is by overexpressing either GFP or or leptin. Again, we proved again the leptin overexpression promotes promotes growth in control conditions or with actinomycin that blocks transcription. You indeed see the leptin overexpression does not promote regenerative growth with actinomycin, suggesting it needs transcription. And then we measure again a transcriptional readout in neurons, phosphoserine 2 on RNA polymerase 2. This is in line with leptin activation that suggesting it needs transcription. And this is, in summary, what I think I've shown you, that leptin binds leptin receptors, activates transcription to sustain axonal regeneration. What I think is happening, this is just a summary of what I told you, but what I think is happening is the leptin basically uncouples, leptin signaling uncouples feeding behavior, because leptin signaling stops you from eating, from axonal regeneration, from a repair mechanism, probably because you need to have some compartmentalization of energy-demanding tasks. But this is, of course, just an hypothesis because I kept on wondering why that is the case. Now, if you think along these lines, you will probably appreciate the last part of my talk. It will be quite easy, I think, for you to follow because, again, it's the same kind of experiments, which is, again, now in this case, focused on peripheral nerve, sciatic nerve crush. But this question we wanted to ask is, over the years, we've done lots of RNA sequencing in different conditions. So now it was natural to think, is there anything underpinning these different conditions that are in blue, pro-regenerative, in red, non-regenerative, across injury conditions, peripheral nerve injury, non-injury conditions, so priming conditions such as environmental enrichment or intermittent fasting, or a combination of environmental enrichment and peripheral nerve injury, versus conditions where regeneration is either strongly reduced with aging or doesn't happen with the central spinal cord injury. And then we perform very simply an enrichment analysis of pathways across these data sets. And what came out is the circadian clock was, you will see now, probably something that is in phase with the regenerative conditions and anti-phase with aging and spinal cord injury. What you can see here is regenerative conditions, and in white, DCA. Non-regenerative conditions, circadian rhythms are at the top of the list. Well, you look, it's quite interesting because we did these things independently, and then after a few years, things come together. Lipid and sterol are up here, and these are leptin-dependent. And leptin aligns the circadian rhythm. So actually, it was nice to see this. That's why I'm telling you, conceptually, things are quite connected. What we did here, we looked at a number of clock core genes into DRG neurons to see whether DRG sensory neurons were following the expected rhythmicity in the clock. I think you all know, familiar with the clock. You all know, right, that there is a circadian-dependent oscillation in signaling in all cells, not only in the central clock. Although in DRG neurons, nobody, I think, had shown it before, probably this is not too unexpected in my view, because all cells have a circadian clock. Just for you as a reminder, this is basic cell biology, but just as a reminder, the clock is a self-regulating oscillatory control system, relies on two genes, clock and protein BMAL, bind to EBOX sequences, activate PER and CRY1, that in reverse inhibit BMAL and clock. So it's a self-sustaining inhibiting mechanism. There are, of course, other genes that contribute, but the only critical, indispensable, non-redundant gene is BMAL1. You will see why subsequent experiments focus on conditional deletion of BMAL1. All right, what Francesco did in the lab with a couple of other people, they did axonal sciatic crush injuries every 4 hours throughout the 24 hours. Then they waited either 1 day or 3 days and measured axonal regeneration. What they found is that at the all different time points, there was not much difference in axonal regeneration, but at Zeitgeber 20, this is a technical terminology, it means 3 a.m., which is 3 p.m. for humans, okay? 3 a.m. for animals. We have maximal axonal regeneration. Then in subsequent experiments, we always use the opposite time, 3 a.m. versus 3 p.m., because of course I couldn't do the whole set for all experiments. We didn't have at the time, you can have these chambers where you can invert the cycles, but we didn't have them, so they actually had to go through the night. All right, so now we know that 3 a.m. enhances axonal regeneration. We did sequencing again from enriched neuronal population at these 2 time points following an injury. And you can see there's a very strong separation. 3 biological replicates at 3 p.m., 4 biological replicates at 3 a.m. And you can see circadian rhythms are not very top, but they are significantly dysregulated. Transcription, phosphorylation, signaling, probably not surprising, but it was very good to see these classes that were differentially regulated. And then you see a number of regeneration-associated genes that are also dysregulated, and a number of core and associated core genes. All right, so this suggested that indeed 3 a.m. had an enhancement in a number of regeneration-associated mechanisms supporting the anatomical data. Next, okay, so before going next, I just want to show you the nice separation of the 2 groups between 3 a.m. and 3 p.m., despite both having an injury. So this was partially surprising, I would say. Again, Fisher t-test with the odds ratio here that I quite like to do because it's a kind of an unbiased, gives you an unbiased, broad view of tendencies in terms of gene expression changes. Of course, you have always more information than you need when you do sequencing. And you see here that a circadian regenerative gain, 3 a.m. versus 3 p.m., is significantly associated with several regenerative conditions. Not all. For example, in rich environment, it does not have injury in itself. But intermittent fasting is also without injury, but it's very strongly associated with circadian rhythm. And we know the circadian rhythm aligns intermittent fasting. I stress this thinking about, in a way, the previous story. So it's nice to see, and there was no association with no regenerative injury. Now, next, what we did, we conditionally deleted BMAL1 in sensory neurons, and then we looked at axonal regeneration after doing the injury at 3 p.m. versus 3 a.m. But basically, what this shows, 2 different ways of measuring it, is that BMAL1 conditional deletion blocks regeneration only when the regeneration is circadian dependent, apparently, at 3 p.m., but not at 3 a.m., suggesting that it is not a general disruption of the molecular transcriptional mechanism, but seems to be circadian specific. And so I think it was nice to see this time-dependent inhibition of axonal regeneration. Next, again, we looked for weeks later when we did injury at 3 a.m. versus 3 p.m. in control versus BMAL1 conditionally deleted animals. And this was the most surprising data, because 4 weeks later, we still have differences in epidermal innervation. 3 a.m. you have more. BMAL1 deletion blocks the benefit. Then we did retrograde tracing to support the data with CTB in distally to the lesion site. It's a very simple experiment, but it's quite useful and robust, where you can count retrograde labeled axons that mark regenerating neurons in the 4 different paradigms, and you see similar results as epidermal innervation, suggesting long-term effects of the circadian clock, which is BMAL dependent on axonal regenerative ability. Here, just to show you the overexpression of classical regeneration-associated genes at 3 a.m. that were also found in the RNA sequencing, but independently from RNA sequencing. These were published before, and these changes are significantly stunted, reduced in case of BMAL1 null animals, conditionally deleted animals. I'm now getting to the end of my story, just show you at the protein level in GFP expressing control or CRE-deleted BMAL1 neurons. You see here the expression of phospho-S6, which is downstream mTOR signaling, or I think it's phospho-CREB here, showing that these important signaling mechanisms at the protein level are also circadian dependent and BMAL1 dependent. The last experiment I want to show you is quite early. I would say it's a, how do you call it, pilot experiment, if you will, at this stage, to prove whether it is possible to use a drug that is known to align the clock and amplify the rhythm by activating certain clock genes. It's been shown in cell lines, in cancer cells, in other conditions, which is called lithium. It's a very well-known drug that, of course, has got multifactorial effects. Interesting, lithium is a GSK3 inhibitor, mainly. GSK3 beta, if you Google it, it's part of the secondary genes that regulate the clock. So the two things are actually connected. And lithium promoted at 3 days following injury some level of axonal regeneration. It's not particularly exciting. That's why I call it like a pilot proof of principle experiment. What was good to see was that in conditionally deleted BMAL1, the lithium effect was blocked. Also, we use CRY1-2. It's a different kind of circadian clock genes. And also, it was blocked. So suggesting, at least in these experimental settings, lithium promoted regeneration in a clock-dependent way. More experiments are needed. And I think probably better specific clock drugs are needed to perhaps use this as a therapeutic intervention. And this is just the summary. I think I told you this. I can just mention this might have implications for restorative interventions, such as timed neurorehabilitation. And as I mentioned, chronoactive drugs could be used. On a less exciting, more prosaic level, this also warns us, although I think we knew it kind of already, that we should really be very careful when we do our experiments, because some of the variability that we see, which is, of course, due to probably many factors, might also be contributed to by doing experiments at very different times. And on the positive side, when the paper will come out, you can have a look at the usual number of supplementary figures. You can see all the different time points. On the positive side, there's a huge time span. There's not so much difference in axonal regeneration. So you can be kind of quite flexible still. And so I'm going to thank. This work is done in collaboration by several people in the lab, but Jessica led the leptin story. Tom, in Rich Environment, was published a few years ago. Now he's in Switzerland. Elizabeth drove the IPA story, Francesco the circadian one. He's now also in Switzerland. I need to thank Dylan Dodd for IPA, collaboration IPA work, Gregoire for some of the enriched environment experiments in rats, and Alexander for measuring IPA, and Matt for some of the bioinformatic analysis. And these are the founders, and yeah, thank you. By the way, I've been reminded, if you're interested in a postdoc, send me an email.
Okay, that was great. Any questions from the audience? Yeah, can find upstairs.
Thank you, Professor, for your talk. And I'm wondering, have you done any functional analysis on the regenerated neurons and see if they are functional and which mode of, for example, the IPA guided or the leptin guided regeneration yielded more functional neurons?
You mean in terms of neurological recovery?
Yeah, yes. Or like
IPA, yes, I showed it to you. IPA enhanced accelerated sensory recovery, at least of some modalities. With leptin, we didn't measure that.
Oh, okay. And have you thought of, for example, a cocktail treatments that combine leptin and IPA for regeneration?
Yes, but I decided not to do it because I prefer to do something else. But that's the honest answer. But yes, it's a good point, but I have to choose. So I decided, to do something different. But it's, of course, a good point. Yeah.
Thank you.
Okay, maybe okay.
Thank you very much for the interesting talk. I want to, so the last part regarding the circadian clock. So the assay of the regeneration has been done 4 weeks later than injury. And then what matters is the time of the injury, right? So it may be, for example, while they are sleeping, like ZD12 is much more capable to regenerate rather than GT8. So which means that like a sleep state or less locomotor activity allows them to regenerate more.
Yeah, yes. It's a very good question that I always get, meaning that it's a good question. And I can answer in a simple way in both cases. So locomotor activity. So I guess you're thinking that animals during the night, they have more locomotor activity, right, compared to the day. So is locomotor activity influencing circadian-dependent regeneration? Very good point. When we did BMAL1 conditional deletion in sensory neurons and we measure locomotor activity, we didn't see any difference. This is one line of evidence that suggests that probably that doesn't make a big difference. Additionally, in the other 3 hours, or no, 4 hours, during the, there are about 12 hours when locomotor activity is quite high for the animals versus the other 12 hours, and the other time points we don't see any difference. So anyways, in conclusion, we don't think locomotor activity has an influence on that. Sleep, it's a different story. We did not sleep deprive the animals, so invert the cycle. It would have been a very good experiment. I was waiting reviewer to ask. They didn't, so I didn't do it. But I would expect that if you, would it be actually a very good experiment, that if you disrupt the cycle, by sleep deprivation, inverting the sleep, then you should not see a 3 a.m. specific increase in axonal regeneration. Maybe at some other time, I guess, or maybe not. So we didn't do that, but it's a very good point.
If it's some kind of sleep dependent or solely circadian clock dependent, what is the actual mechanism you can think of? I was thinking about to try to connect leptin story, but perhaps because of the like feeding state, circadian clock will be reset.
Yes.
Maybe leptin and the feeding and regeneration or sleep even, maybe all coupled.
I agree 100%. Because, and again, it was nice because for us, independently, we developed kind of these stories, and then my conclusion earlier after leptin is based also upon the circadian work. So basically, yes, intermittent fasting, promoting regeneration, I do think has a circadian clock dependent mechanism. We should do intermittent fasting, BMAL1 deletion, I guess, right? And see if indeed that is the case to prove this, the point. But yes, in summary, I agree with you. Problem is feeding, time of the day, and sleep. They all contribute to control, I think, tissue repair, probably broadly, because I cannot imagine that nature just focuses on nerve regeneration, right? So, yeah, and I think it makes sense also because anything you do in your life is quite compartmentalized. You don't typically go to the bathroom, read and eat at the same time, and talk at the same time, and and so on. You can do a couple of things maybe, but not all together. So things need to be compartmentalized in an energy-dependent, probably manner. And also, I think that's important to a compartmentalization of tasks to have enough time to escape from predators, or I don't know, there are a number we can discuss at length. I will stop here, but I think it's quite interesting, broadly speaking.
Interesting. Thank you very much.
Okay.
Hi, thanks for the very nice talk. And I have 2 naive questions. The first one is, could this intermediate fasting affect the microbiota in the gut? The second one is, how does IPA affect the neutrophils' functions? Thank you.
Sorry, I didn't understand the last question. Which function?
Neutrophil.
Neutrophil, okay. First question is very easy, so we published that. So it's, again, supplementary figure 12. And we did look at the gut microbiome following intermittent fasting. There are changes in the gut microbiome. They're not dramatic changes, but there are significant changes in specific bacterial species, including the ones that belong to Clostridium, that Clostridium sporogens belong to. So you can have a look, if you like. And the second question, neutrophil activity. Okay, so we did not study neutrophil mechanism in a systematic way following IPA. However, what we found is the neutrophil-dependent interferon gamma production enhances several regeneration-associated genes that depend on interferon receptor expression in sensory neurons, and this is IPA-dependent.
Did you check other types of immune cells, like T cells, and can also secrete interferon gamma?
Yes. We did check, again, we looked at all different types, the major types of immune cells, innate adaptive immunity. We looked at their presence at the injury site and the numbers, and at the DRG site, at the cell body site. Unlike neutrophils, they increased their numbers in the DRGs. We didn't see any differences. However, that does not mean that maybe their signaling might still be affected. But those are the experiments we did. We didn't see, unlike neutrophils, any changes in their presence and general activation state in the lesion site and the dorsal part.
Okay, maybe I have a couple questions. So the leptin signal, leptin part, so you have to give using AAV, then you can see the very strong regeneration. But how about the systematic leptin? Would that have reached the level to see a significant regeneration?
Yeah, so we gave leptin, and I've shown that systemically, IP. And that promotes regeneration. We looked at regeneration of the sciatic nerve injury. Yeah. We did not do that experiment at the spinal cord injury. The main reason is because I wanted to look at specific mechanism. And if you do give leptin, of course, it's a good experiment. It's just maybe a more translational experiment, yes. If you give systemic leptin after spinal cord injury, it's difficult to understand what is going on. Actually, leptin is being given after spinal cord injury, by the way, in the past, and they showed some neuroprotection, some positive effect. I don't remember anymore. It was not a particularly exciting paper anyway, but there is some literature there. And that, in a way, was for us a disincentive to do it. And also we understood that, we would have probably not learned too much. And plus, I think if you go down that route, it's a lot of work because you have to do ideally different concentrations because it's difficult to control. And But of course, it wouldn't be bad.
So the BM1 knockout you showed, you only did it in a conditional knockout, right, in DRG, not a systematic knockout. What happened to systematic knockout?
BMAL1?
Yeah, BM1. BMAL1.
So in BMAL1...
the circadian has been disrupted.
It disrupted everywhere.
Yeah.
So we didn't do that because I think that probably would not give too much information. But we thought about, but again, we didn't do it. Nobody asked. It would be interesting to do BMAL1 conditional deletion, take deleted mice in the central clock and see if you disrupt, maybe that's what you were thinking about. So if you disrupt the central clock only and not the peripheral clock, do we see a difference? And so suggesting maybe more also contribution of metabolic.
Rather than within
that would
the
be
cell,
quite a cool experiment, and
it would be interesting.
Yes.
Yeah. The last part, actually, the last question is it's amazing. You showed if you do the lesion at, let's say, 3 a.m., right? Yeah. But you see the effect like last 4 weeks. So what exactly happens? Let's say is because of right after the injury, those initiation of axons very fast. Let's say because usually we know. In vivo, take about a day, right, for the axons to start to initiate to grow. Seems like in those mutant, in the time you do it, they can initiate faster. Would that actually the major reason why you can see a functional recovery even later on? I mean, or some other reasons, right?
Yeah, I don't know. That's a good point. But to know that, one should do a bit of a time course to see if they
continue have this difference or only at the very initial.
We know that at 24 hours they already have an advantage, and at 3 days they keep on having an advantage. But as you know, then in the periphery, it's later on. If you measure the axons, become too dense. It's difficult to see later on. But we know about those 2 time points, so it's possible they have an advantage that the others don't catch up with. In terms of molecular mechanism, what I can imagine is that sensory neurons are primed. Already time-dependent, time-tuned is primed at 3 a.m. And then injury comes, and the signals come systemically, and retrograde signals come to the dorsal ganglia, and that priming mechanism that probably relies on epigenetic, I think, changes that we didn't study systematically allows then the long-term advantage. That's how I can imagine it.
Right.
Yeah.
So because the stat 3 is high, right, the time, right? So like stronger and faster transcriptional change to sustain a relatively longer.
Yes, I think that's the most likely explanation. Yeah.
It would be nice to look at what happened to the injury. That's the injury signal we know is also very critical. So at the time of injury, whether locally there's some kind of change.
We looked at that, of course, and that will be published supplementary figure where, again, yes, we looked at all the different the classical immune cells and their activation state after injury at the different times. We couldn't see a difference. Oh. But I'm also aware and that if you look at the injury site, it's an extremely rich with immune cells. So I think can be a bit tricky to observe differences at the region site, anatomically, morphologically, because there are so many, because it's so dense, with those cells. So a better way that we didn't do, I think, would be to sequence, the different cell populations at different time points and see whether there's a difference. I would be surprised if there is no difference. Yeah, so there must be a difference. Regardless, when we conditionally delete BMAL1 in sensory neurons, we block the phenotype. Of course, that does not exclude that there is an upstream signaling communicating with neurons, and then we block it by BMAL1 conditional deletion because that's a core clock mechanism. So I think actually the two mechanisms are not mutually exclusive, despite what we will show in the supplementary that there's no difference in immune cells. Yeah.
Okay, any other questions? Okay, go ahead.
May I add a question? So I'm curious to know whether the regeneration speed itself is also associated with the circadian that you have described. Like, for example, does it grow faster in the morning but just stop or just pause in the afternoon? Is there a correlation between these speed and circadian itself?
Yeah, well, typically when you look at sensory neurons in the periphery after, say, a crash, you look at speed, meaning that they grow slower or faster. Okay, so then you can have sometimes the density is potentially another factor, but typically what people have shown is the speed. So I think what we are seeing in our data is actually different speeds at different times.
Okay, but I think that may be an overall result for multiple days that you have watched. So do you carefully look at the specific time points, like just about half a day, what will be the speed change?
Yeah, so you think that's a speed difference?
I'm just curious to know.
I think so, yeah, I think so. I think it is a speed difference, yeah.
Okay, thank
difference
you.
in speed, yeah.
You need to look at basically slope, right? So that will give you.
Yeah, thank you.
You could, actually, yes. I mean, we could potentially graph, I guess, yes, the 3 time points that we have, in a way, in a slope kind of, yeah. We didn't do it, but I guess the data is there. Yeah, one can do it, yeah.
I have a question about the BMAL1. As you mentioned, every cell has a circadian. Is BMAL1 is DRG specific, or do you check the BMAL1 work in any other cell types or any other neurons in other regions? Thank you.
So BMAL1 is ubiquitously expressed. But in this paper, I didn't have time to show you, you can look, I think we did a specific experiment where we cultured DRG neurons, and then we use a reporter with luciferase, okay? So a reporter gene, which is a pair, anyway, it doesn't matter, which is a target of BMAL, okay, BMAL1. And we see that there is a specific oscillation throughout the day of the signal in DRG neurons. Now, when we delete BMAL, this oscillation is blocked in DRG neurons. Then we do a rescue experiment. We put BMAL back in, and the oscillation, circadian oscillation, is restored. Plus, the in vivo experiment that I show you with conditional deletion of BMAL in neurons show that our mechanism depends upon BMAL1 in sensory neurons. Okay? But as now discussed with Kai, we don't know whether if you disrupt BMAL1, which again is expressed in all cells, only in the central clock, which is in specific nuclei in the brain, also disrupt the axonal regenerative ability of sensory neurons. We didn't do that experiment. We don't know that, but what we do know is the disruption of BMAL1 specifically in sensory neurons is sufficient to disrupt the clock-dependent regenerative ability. Does it make sense?
I think her approach maybe another aspect. I say, what if to the spinal cord injury or the, for example, optic nerve, you would expect similar kind of.
Yes, yes. So spinal cord injury does not regenerate typically. So we didn't do BMAL1 condition deletion after central spinal cord injury because it is a no regenerative condition. However, one could do, for example, so-called conditioning lesion, where you do have some improvement in sensory axonal regeneration, or actually now that I know, hopefully, one could also do leptin. Would be a great experiment. Actually, would be to do leptin overexpression in the G neurons that we also know aligns the clock. God, when I say it, I hope, the reviewers, this is probably, the other one will not ask me. But that would be actually a great experiment, right? Leptin overexpression in sensory neurons and condition delete BMAL1 and see if that disrupts regeneration in the spinal cord. Because, anyways, we didn't do it. But that would be, I guess, probably the best experiment.
Okay, any other? Great. I mean, so this is, Simone gave us a very comprehensive understanding how not only the neuron itself and also not only the so-called injury inhibitor, there's a very, environmental interaction, including immune system or other factors interact with the neurons and affect the regeneration. Okay, let's give another round of applause. And after that.
Thank you.